This method can help answer key questions about the stem cell-based treatment of peripheral neuropathies, particularly for equine recurrent laryngeal neuropathy. The main advantages of this technique are that the stem cell harvest is minimally invasive and that the injection method can also be transferred to other peripheral neuropathies.
The implications of this technique extend toward the study of other cell types, as the stem cells can be differentiated into other cells of interest before the injection.
Visual demonstration of this method is critical, as the video endoscopic control of the laryngeal movements and the manipulation of the nerve stimulator techniques can be difficult to learn.
Identify the sample site in the long head of the triceps muscle by visual inspection and palpation about midline between the point of the elbow and the point of the shoulder. Use electric clippers to shave an approximately 2 by 2 cm area and apply a surgical scrub consisting of polyiodide liquid soap and alcohol compresses for 1 minute to the exposed skin. Using a 25-gauge needle, subcutaneously inject 1 milliliter of 2% lidocaine solution into the center of the disinfected area. After disinfecting with clean antibacterial hand soap and donning sterile gloves, place a semi-automatic 14-gauge biopsy needle on a disposable sterile drape and pull on the spring mechanism to arm the needle. Assemble a cannula and obturator in the locked position to form a trocar, and introduce the trocar perpendicularly through the desensitized skin, approximately 1.5 centimeters into the underlying muscle.